So I compiled the 3 world leaders in schizophrenia research: GE Neuro with their human endogenous retrovirus type W research, Stanley Medical Research Institute with their brain structure and repurposed drug database research, as well as being a patron to GE Neuro and the University of Oxford with their research to anti-NMDA encephalitis. Coming up with my own hypothesis, which I call the pregnenolone hypothesis. While it is more to do with cholesterol, it initially had to do with pregnenolone. I'm going to give a brief introduction to the neurology of the mechanics in this hypothesis. I had to pull some of the models of Wikipedia explaining steroidogenesis because they are less dense than the alternatives. 10% of people with schizophrenia actually have anti-NMDA encephalitis, anti-VKG-C encephalitis, which is voltage-gated calcium channel antibodies, voltage-gated potassium channel antibodies. To my knowledge, is slightly less common, so let's assume that makes up 5%. This leaves 85%, which according to Belinda Lennox from the University of Oxford, carrying antibodies that target NMDA. 85% of these people will still carry unknown antibodies, but haven't unidentified pathology and significance. I'm going to focus on the 85%. I'm going to call these atypical anti-NMDA antibodies. Firstly, what is important is that in 100% of cases of the sample taken in Belinda's case study, the immune system attacks NMDAR. This is important because it strongly suggests an autoimmune disorder. NMDA is an excitotoxin and kills nerve cells. Overactivity can be caused by a variety of factors. So I've got this study by Belinda Lennox, and it's explaining that the link is not clear with these other antibodies, but that everyone with schizophrenia in their case study carried some sort of anti-NMDA antibody, whether it be the 2 known ones or the 85% unknown ones. Now I'm going on to the next figure. This is to do with the steroidogenesis cascade, which means the chemicals which are synthesized into neuroactive steroids, which ends in a repeating loop which starts over and over again to make more steroids. This figure and the next figure have slightly different interpretations of the steroidogenesis pathways because of disagreements about the conditions for synthesis. Putting that aside, you can see in the second figure how allopregnanolone is produced through preg-prog-allo. Neuroactive steroids are important in neuropsychiatric autoimmune disorders as well as in any autoimmune disease, covering multiple roles from regulating inflammatory response to estrogen being required for proper immune response to bacterial and viral pathogens, at least in the female brain. So going back to the first figure, you can see it starts at the very top left. Cholesterol side chain cleavage enzyme gets synthesized into pregnenolone, which gets synthesized into progesterone, and it ends up down the bottom with the sex steroids, testosterone and estrogen, and glucocorticoids and mineralocorticoids, which are important in regulating inflammatory response as well as salt levels in the blood, which is important for the immune system. Neuroactive steroids are essentially the chemicals which control the immune system from the brain, and these are the systems which break down in serious neuropsychiatric disorders. Back onto the second model, you can see this is one that covers allopregnanolone from cholesterol, pregnenolone, progesterone, DHP, and allopregnanolone. I'll explain the significance of allopregnanolone later. Moving on to the next figure. This is what's known as the cholesterol connection. What happens is that viral pathogens, microbes, will pass a cell wall and they'll be enclosed by the plasma of cholesterol, which will protect them from being targeted by the immune system. You can see all through these red gaps in the cell wall that are cholesterol eventually encapsulates the pathogens, which then move around the body freely on cholesterol lipid rafts. This is important, and I'll talk about why later. Before I move on to the main explanation, I think it's also important to cover how receptors work. And so far I've covered how steroidogenesis works, I've covered how cholesterol transports pathogens around the body, and before I move on to the NMDA receptor specifically, I'm going to target some of the allostergic functions of that receptor, and in broad, any receptor. In biochemistry, allostergic regulation is regulation of an enzyme by the binding of an effector molecule at a site other than the enzyme's active site, often resulting in a change of size, shape, and protein dynamics. This enhances the protein's activity and is referred to as allosteric activators, whereas that decrease the protein's activities are called allosteric inhibitors. So you've got activators and you've got inhibitors. A good example of this is psychotropic drugs. There is enzymes which are responsible for blood plasma metabolism of psychotropic drugs, and taking an inhibitor such as St. John's worts and some herbal remedies can compromise the effectiveness of the drugs because the blood plasma metabolism changes, which means that they no longer break down the chemical as they were before. Whereas there's also some substrates which increase the concentration of the drugs, though I'm unfamiliar with any of them off the top of my head. Allostergic regulators are a natural example of control loops, such as feedback from downstream products. Long-range allostergy is especially important in cell signaling. Allostergic regulation is also particularly important in the cell's ability to adjust enzyme activity. Allostergic modulation occurs when an effector binds to the allostergic site, also known as regulatory site, of an enzyme and alters the enzyme activity. Allostergic modulators are designed to fit the allostergic site and cause conformational change of the enzyme, which basically means that the molecule starts doing something different that it was programmed to do when the allostergic site was activated. So, in the case of psychotropic drugs, for example, it could be to make more or less of the drug, depending on the enzymes responsible for blood plasma metabolism. In contrast to typical drugs, modulators are not competitive inhibitors, which means that they don't compete at the site with other drugs, but that's not really important for this example, so I won't go into it further. They can be positive, activating, causing an increase in the enzyme activity, or negative, inhibiting, causing a decrease in the enzyme activity. As you can clearly see from the model here, up top you've got allostergic activation, and down the bottom you've got allostergic inhibition. So you've got the catalytic binding area across from the substrate and the cofacting binding area. And if I remember it correctly from chemistry, if a substrate binds with a catalytic binding area, then it will produce the opposite, which is an inhibitor. Positive allostergic modulation, also known as allostergic activation, occurs when binding one of the ligands enhances attraction between substrate molecules and other binding sites. So that's what I just explained. It's to do with inhibition. Negative allostergic modulation, also known as allostergic inhibition, occurs when the binding of one of the ligands decreases the affinity for substrate at the active sites, meaning that the chemicals responsible to increase enzyme activity become less available. Moving on to the next figure. This is an activated NMDAR receptor. So as you can see, you've got the glycine binding site, you've got the glutamate binding site, and you've got the allostergic binding site, which can be either positive or negative. The NMDA receptor is activated when glutamate and glycine or D-serine bind to it, and when activated it allows positively charged ions to flow through the cell membrane. This is the enzyme that I was talking about. So the NMDA receptor is very important for controlling synaptic plasticity and memory functions. Think of it as one of the master keys to the brain because it's very, very important in signaling. The NMDA is a specific type of glutamate receptor, which primarily makes up the prefrontal cortex, the temporal lobes, and is dopamine-based. As you can see on the figure, you've got the calcium channel, and as not demonstrated on the figure, you've also got the potassium ion channel. And both of them flow through there at a healthy rate if the glycine binding site, the allostergic binding site, and the glutamate binding site are all functioning correctly. The NMDA receptor plays an important role in synaptic plasticity and synaptic formation, underlying memory, learning, and neural networks during development of the central nervous system. Overactivation of the receptor, causing an influx of those calcium and potassium ions, can lead to excitotoxicity, which kills nerve cells and has been implicated in several neurodegenerative disorders. Blocking of NMDA receptors could, in theory, be useful for treating such diseases. However, hyperfunction of the NMDA receptors could possibly be involved in a worsening of states if it was to directly treat the receptor site, because the main problem is the utilization of NMDA receptor antagonists for neuroprotection is that the normal actions of NMDA don't function properly. Successful clinical application of NMDA antagonists would need to block the excessive activation without interference with its normal function. Therefore, the best way to approach this is to identify the cause of overactivation and treat the offending agent rather than target the receptor site itself. Okay, this is my model that I've drawn up for the pregnenolone hypothesis. This is the primary model, which means that it's primary to the cholesterol connection, and there's a direct link between the chain of events, whereas there's a secondary model that I'll explain later. Pathogens infiltrate cholesterol through the plasma wall and hide inside the cholesterol connection. That leads to cholesterol-carrying pathogens, which are transported to the NMDA receptor, positive allostergic binding site. This can lead to two things: sodium channel excitotoxicity, which is associated in major mood disorders, and calcium channel excitotoxicity, which is associated in psychotic disorders. So it may just be that when people develop serious neuropsychiatric disorders, if it's one of those two or one of one of the two or both, in a case of schizoaffective disorder or psychosis with mood symptoms, or mood disorder with psychotic symptoms, is that it's to do with brain structure and stresses when it comes down to the NMDA receptor malfunctioning. Now, there's three things that could happen after the cholesterol is transported to the NMDA receptor. First of all, the brain tries to close the NMDA receptor because of excitotoxicity. Second of all, the brain tries to kill NMDAR because it no longer recognizes it as being functional or part of the brain. Or third of all, the brain tries to kill the pathogens, which have been transported in those cholesterol lipid rasps all the way to the glutamate sites in the brain. Now, all three of these could result in atypical anti-NMDA antibodies being produced. Or it could be a combination of the three, depending on a variety of subsets of different people. I'm going to move on to the secondary model now. This is the secondary model, which is less relevant to the cholesterol connection. It basically means that neuroactive steroids, which is the brain's immune system, and NMDAR just destroy each other but indirectly until their function wears down and they become dysfunctional. So on the far left, I've got low pregnenolone. That causes a downstream cascade. Low pregnenolone affects NMDR. NMDR has poor signaling. This starts an inflammatory cascade because it's neurotoxic and is important in a variety of functions for the immune system to function properly. And when NMDA signaling function breaks down, that results in low pregnenolone because the immune system isn't working properly. And that just goes in a loop over and over and over again before pregnenolone levels lower, which is being exhibited in schizophrenia, as well as a variety of other neuropsychiatric disorders, and impaired NMDAR signaling, which has also been implicated. And both of those things could eventually trigger atypical anti-NMDA antibodies. So it's a secondary model because not just because the cholesterol connection is less relevant, but also because it could be either pregnenolone or NMDA that trigger the production of atypical anti-NMDA antibodies, and they aren't necessarily directly connected to each other, which is steroidogenesis and NMDAR. Okay, I'm going to go through some afterthoughts and some questions maybe to ask about the model, which I demonstrated in the last two figures. First of all, it's been shown that cholesterol-lipid raft mechanisms, or the cholesterol connection, has been shown to alter cholesterol synthesis pathways. Prog-allo, which is pregnenolone, progesterone, allopregnenolone, is wired early in schizophrenia. This is seen to be one of the first structural or functional changes that occur at the beginning, if not the beginning, of the disease. Both of these could be structural brain deficits that predispose steroidogenesis malfunctioning, or a consequence of the mechanisms which could potentially encourage further degradation of NMDAR and function and steroidogenesis synthesis. In the primary model, if the cholesterol connection triggers a neuropsychiatric autoimmune response, it is possible that it goes on for long enough this could rewire the immune system, i.e. steroidogenesis, to attack the brain. Poor brain structure from genetics could predispose people to this autoimmune response. Before or after the cholesterol raft mechanism carrying pathogens through the blood-brain barrier and rewiring of steroidogenesis and cholesterol synthesis pathways. Pregnenolone sulfate is low in schizophrenia. This is important because it's a weak allostergic modulator of NMDA, but also because of other chemicals that are produced in steroidogenesis cascade, such as testosterone and estrogen, which are important in the role of cognition, linking steroidogenesis to psychological stress models of mental illness and neuropsychiatric disorders, glucocorticoids, and their role to mediate inflammation response. Glucocorticoids, with their role to mediate the inflammation response, are also dysfunctional because the steroidogenesis cascade is dysfunctional. In theory, abnormal wiring of the steroidogenesis cascade and cholesterol synthesis pathways could ultimately affect the balance of NMDAR positive and negative allostergic modulators, potentially causing a catalyst reaction even more complex than initially thought. What's important is that not only is pregnenolone sulfate, the derivative of pregnenolone, a weak positive allostergic modulator of NMDA, but so is cholesterol and pregnenolone. Okay, questions. I just finished with the afterthoughts. This is some pathologies to look into to explain the exact mechanism or identify several subsets. The first one is human endogenous type W. This is important because 60% of people with schizophrenia or bipolar disorder seem to carry this retrovirus. It was implicated in multiple sclerosis and is now being undertaken as a research project to the only drug target which is shown to have an improvement in the condition, although further funding has not gone ahead at this point. Pregnenolone supplementation has been shown to be beneficial in some people with MS, so it's possible that this similarity in neuropsychiatric disorders could have a similar origin. Brain stroke antibody mechanisms. After a brain stroke, the area affected by the brain stroke stops receiving sugar and oxygen from the blood. A similar mechanism could possibly kickstart an autoimmune disorder after neuron death of systems related to NMDA or neuroactive steroids, depending on when the neuron death is. Anything as small as head trauma can cause neuron death. Systemic lupus erythematosus. Lupus is very important because it's an autoimmune disorder that mimics schizophrenia, and looking at the pathology of that, how that works could reveal some underlying knowledge, given that it causes the anti-NMDA encephalitis. The known one makes up 10% of schizophrenia. So the pathology could still be relevant to the other 85%, which is neither anti-NMDA encephalitis or anti-VKGC antibodies either. Look at all known autoimmune disorders. Look at all types of immune stress, ranging from psychosocial stress to hypothermia response. Can anti-NMDA encephalitis antibodies or anti-VKGC antibodies develop into atypical anti-NMDA antibodies, or the other way around? This is an interesting question. All known diseases that use lipid rafts or similar mechanisms could reveal several different subsets of the cholesterol connection using lipid rafts to transport diseases to the NMDA receptor. Eli Lilly, the creator of olanzapine and several other psychotropic drugs, tried to produce an allostergic modulator compound as well as a novel antipsychotic called LY404039. However, after initial testing, they withdrew the production further from development. In 2013, this can be explained similar to SMRI's pregnenolone oral supplementation, that Stanley Medical Research Institute's repurposed drug database, where they repurposed drugs for the use of schizophrenia, which are initially used for other things. While some of the levels of neuroactive steroids are normalized in both cases, the cholesterol transporting pathogens are not removed. Neither is autoimmune response for all the wiring deficits. Anti-cholesterol drugs are shown to have antimicrobial properties because they stop the cholesterol raft mechanism by destroying it and letting out the pathogen to die. This may be an interesting pathology to look into for potential treatment avenues of novel antipsychotics or other sorts of treatment. Essentially, in a pregnenolone hypothesis, there are only two drug targets that need to be focused: the NMDA, the target of the immune system, and neuroactive steroids, the neuroactive immune system. Both clozapine and olanzapine, the two most clinically significant efficacious antipsychotics, are the only two drugs of their class to raise brain pregnenolone levels in both humans and rodents. The anti-NMDAR is shown to polymorph in late-stage bipolar disorder. So, it's clearly a very important drug target, but as reiterated earlier, it may be more important not to treat it as a drug target, but rather the target of a secondary autoimmune disorder for the best results. Now I'm going to enclose my bibliography, which you can refer to resources for research to back up my assertions. While I go through the bibliography slides, I'm going to clarify some information on the earlier two models of the NMDA receptor and allostergic modulation. A ligand is any molecule that binds what is usually a large molecule and changes its function. An example of a ligand of NMDA is pregnenolone sulfate. Negative allostergic modulation leads to allostergic inhibition, which leads to limited bioavailability of the ligand in question at the binding site with less of a reaction. Positive allostergic modulation leads to an increased reaction and more of a target chemical produced. As NMDA is both ionotropic and glutamate-based, I am unsure of the effects of the relationship of modulation to excitotoxicity. Neither of these is important to excitotoxicity, however, as my model focuses on the upset of the natural ratio of allostergic modulators to compromise steroidogenesis synthesis pathways and cholesterol acting as a raft to carry pathogens past the blood-brain barrier and possibly even to the NMDA receptor.